Hi, in this video I have prepared two questions. One is a multiple choice question and another one true-false question. And as usual, I recommend you to stop video here, read the questions, choose the correct answer, and when you would be ready, you can run video again and you can compare your answer with my answer and explanation. So here's the first question. Most genes in prokaryotes and eukaryotes are regulated primarily at which level of the expression? And here is the 5 answers to choose from, and before I will give you an answer, let me show you the central dogma of molecular biology. This is a flow of information from DNA to protein. So at the first step we have double-stranded DNA, so this is DNA, and with the first step message RNA would be made, and we call this process transcription. So this is message RNA, and with the next step to this message RNA would be processed, and would two caps would be added. This is going to be 5' end cap, and also 3' end polyadenyl tail would be added. So we call this processing, and in prokaryotes there is no such step, because in prokaryotes message RNA would be instantly processed into the protein. But in eukaryote, message RNA have to leave the nucleus first. So for the stability of the message RNA, it would be processed, and of course introns would be deleted. And with the next step, this message RNA would be used in the process of translation to make a protein. So this is going to be polypeptide chain, and this polypeptide chain also would be processed in order to make a three-dimensional form, so it would fold. And also there is one more step. When we have usually a protein in the organism, it doesn't work alone. Usually it needs to aggregate with other proteins to become active. And consider, for example, such cell machinery as ribosome that consists of 50 different proteins and about the same number of RNAs that we call ribosomal RNA. So this is how information flows from DNA and into the protein. So as you see, we have at least 1, 2, 3, 4, and 5 steps here. So now you have the whole picture, and now if you think at which step it would make more sense to, for example, interrupt production of the protein. It can be made here, and protein wouldn't be made if we interrupt it here. But of course, nature doesn't like excessive work to do, and of course the primary regulation would be made here at the beginning, at the stage that we call transcription. So all the rest would be translation, step number 3. We can cross out protein activation. It can be step 4 or 5. This would be the last steps, so we can cross out them. RNA processing, that is step 2 and 3. So message RNA stability, also step 2. So we can cross out the steps, and the most important step to regulate message RNA stability, production, and processing, and production of the protein would be at the first step. This is going to be transcription. So the correct answer would be A. And next statement, frameshift mutations generally have more drastic effects on the phenotype than do substitutions. And we have just two choices, true or false, and imagine that this is DNA. So this is DNA, single strand, and this can be, for example, coding strand of the DNA, and it's going to be the same as message RNA, the same code. So on the DNA here we would have different bases, for example adenine, guanine, cytosine, guanine, thymine, adenine, guanine, cytosine, guanine, and guanine, for example. And every 3 bases here code for 1 out of 20 proteins. And imagine that here this cytosine would be substituted with different base, for example guanine or thymine or adenine. This might lead to that this trinucleotide or codon would code for the different protein, but the rest would be the same. And because this is third nucleotide in this trinucleotide, usually this, when we change or substitute this base with a different base, still this would code for the same protein, because we call this wobble position, and usually up to 6 different combination may code for the same protein, but all the changes usually tolerated at the last position. If we change at the second position or at the first position, this would definitely mean different protein. So as you see, substitutions sometimes may lead to new protein, sometimes not, but it doesn't affect the rest of the proteins in the protein chain. But if we delete, for example, one of these bases here, for example this base, we are going to get a frameshift. So new trinucleotide would be this one, the second would be this one, and the third would be this one. As you see, this is completely new sequence, and we not only change this sequence that code for the first protein in the polypeptide, but also all the rest would be changed. So the whole protein would be changed, and the function of such protein also would be changed, or this would be non-functional protein altogether. And as you see, frameshift mutations generally have more drastic effects on the phenotype than do substitutions. And this is true, and this is answer A. And this is all for today. Thank you for your attention. Please subscribe for more new videos that I post almost every day. Thumbs up if you like this video. Please write your comments, questions if you have any, and see you in the next video. Goodbye.